Hey guys, it's Miss Huber, and this is the second part of your notes here. So this is going to be chapter 2 notes, and I've consolidated this down. It's part A and part B kind of smashed together. I made this slide first. So the first part basically covers chemistry basics. So it talks about atoms, it talks about different states of matter, talks about mixtures and solutions. This is all stuff that you should have already taken because you guys are upperclassmen. You should have had biology and chemistry already. So I'm not going to cover basic chemistry. So it tells you that basic chemistry starts on slide 4. And then the things that you are expected to know all that information, but the things that are going to be maybe new to you or the things that we're going to be focusing on for this particular exam, you're going to need to know the things that start on slide 75 and on. Okay, it's kind of a large presentation, but it does have a lot of like reiteration to it. So I'm just going to tell you this here is like the beginning of like chemistry. So it talks about basic chemistry and biochemistry, goes through to talk about different states of matter, talks about energy, kinetic and potential energy, different forms of energy that we have, energy conversions. We talk about elements and atoms and all of the parts of an atom, talks about the charges of all those different parts, talks about valence electrons, talks about bonding, isotopes, things like that. Like I said, all of this is stuff that you should be familiar with from your general chemistry classes. And if you don't remember it, please feel free to go through and read all of this, read the textbook for sure. The different types of bonds are always really important. Like I said, this is all basic chemistry, and it will turn into our biochemistry. I put in a very large page here for you. So biochemistry starts here. So everything else is considered general chemistry. You are responsible for it, and it is going to be a foundation for what we're about to talk about. But today we're going to focus on biochemistry. So properties of water and biochemistry. So biochemistry is basically a blend of living chemicals, right? Bio, life, chemistry, chemicals. So these are all things that compose living or organic materials. Inorganic compounds are just things that do not contain carbon. Some examples are water, like H2O. There's no C in there. It doesn't contain any carbon. Organic compounds are things that contain carbon. So if you talk about like, oh, we only buy organic. Well, everything that you eat is organic because it's alive. It contains carbon, right? Both of these things are essential for life. Water is one of the most important things for life, and it has certain characteristics that make it a very special solution. So we have high heat capacity, high heat of vaporization. It is a polar solvent, and it has a lot of reactivity with different salts and things like that. And it also acts as cushioning in our body. And we talked a little bit about that in our first set of notes, but it's a really important property to make sure that all of our organs are protected. So water has a high heat capacity. So what does that mean? It's the ability to absorb and release heat with little temperature change. So essentially, this prevents sudden changes in temperature. So water is able to moderate its temperature quite well. It also has a high heat of vaporization. So this is the amount of heat that needs to be put into a substance in order for it to turn into a gas. So like water needs to be heated to turn into steam. So a lot of heat needs to be applied in order for water to turn into steam, which again, helps prevent sudden changes in temperature, but it also acts as a useful cooling mechanism, like when you sweat on your skin, right? When the water is evaporated off of the surface of your skin, it leaves behind a cooling sensation. So it's called evaporative cooling when we experience cooling because we're sweating. Water is also polar. It has a slightly negative side and a slightly positive side. So the oxygen carries a slightly negative charge, and then the two hydrogens carry a slightly positive charge. So it's considered polar because it has both regions. And there's a saying in chemistry that like dissolves like. So polar solvents will dissolve polar substances. Here's an example of that. So we have salts, sodium chloride in here, and it's NaCl. So because it is polar, it has positive and negative ends to it that you can see in the picture. It's going to dissociate or dissolve in water because water is also polar. Polar things dissolve in polar solvents, like water. Water is also reactive. So it's part of hydrolytic and dehydration synthesis reactions. So hydrolysis means to break with water, and dehydration synthesis means to join together, removing water. So these are the two different types of reactions that help us build or break molecules, and we need water to do both of them. And our body uses these types of reactions for almost everything. And then we talked about cushioning. So it helps to protect your organs from physical trauma. Like cerebrospinal fluid cushions your nervous system, right? Your nervous system, especially as you get older, is very, very vital because it doesn't go through a mitotic division anymore. So if you have damage to any of these organs, then it's permanent. So it's very important that we have a cushioning mechanism in order to make sure that they're not just bouncing around on our bones and getting damaged all the time. That's why we have water and cushioning. Salts are important in the body. They act as electrolytes. They're ionic compounds that dissociate into water because, again, they're polar. Like I said, they're called electrolytes. Some of the important ones are sodium, potassium, calcium, and iron in the body, and they're really important for homeostasis because you need those vital salts in order to complete some of the chemical reactions in order to, maintain homeostasis and survive, right? So we need salts to help us do that. We also are going to talk about acids and bases here. So we're talking about pH. Acids and bases are both electrolytes. They ionize and dissociate in water. So acids are considered proton donors. They release hydrogen. So an acid is a substance that contains a lot of hydrogen. And then on the flip side, bases are proton acceptors. They pick up the H plus, so the hydrogen ions. So for example, we have sodium hydroxide here. It dissociates in water. And we have a very high concentration of hydroxide in our bases. So pH is just a measurement of how much hydrogen is in a solution. So if it has a lot of hydrogen in the solution, it's going to be acidic. If it has little and it has a lot of hydroxide in contrast, it's going to be basic. So there's a pH scale, and this talks about that. So if we have anything from, as it says here, 0 to 6.99, so anything that is less than 7 is considered an acid. If it is 7, it is halfway in between 0 and 14 or 1 and 14, depending on your textbooks. 7 is neutral. It's exactly in the middle. It has an equal amount of hydrogen and hydroxide. And our alkaline bases here, so alkaline and basic are the same thing. This is going to be anything that is over 7 is going to be a basic solution. You might see it's popular in grocery stores, basic water is on sale a lot. It's water that has a pH of 8.8 instead of 7. And we talked about this in class, but drinking too much of it can cause damage to your body. So drink in moderation. So here's an example of what the overall scale looks like. So you have your neutral there in the middle, and as you increase in acidity, you're decreasing on the pH scale, and as you increase in your alkaline properties, then you are going to get a bigger pH number. So 14 is the largest, it's the most basic. 1 or 0, like I said, depending on your textbook, is going to be the smallest, that's the most acidic. We also have these things called neutralization reactions where acids and bases are mixed together because they kind of undo each other. So this is an example of that here. We also have buffers. Buffers are going to be solutions that tend to resist changes in pH, which are really important because our body is full of enzymes and enzymes only function at certain pHs. So if the pH is changed, then those enzymes won't function. So it's important that we have buffers to help resist changes in the pH. I'm going to skip through some of this. Polymers and monomers are important for you to understand because we're about to talk about the macromolecules of life. Polymers are many pieces put together. That's what polymer means. And monomer is a building block. It is literally one piece. So we make polymers through dehydration synthesis, and we make monomers through hydrolysis. And again, I talked about those two reactions. We talked about water. So here's some examples here. Hydrolysis, literally you're adding in water to have one molecule break apart into two. And the opposite, up at the top, it's kind of hard to read there, but dehydration synthesis is when we have two molecules, monomer 1 and 2, that we are removing a water molecule from in order to combine them. We're making a polymer there. So our first macromolecule we're going to talk about is a carbohydrate. Carbohydrates are sugars, carbs, sugars, starches. Okay, they contain CHO. I call it CHO. So We're going to learn CHO, and CHON-P. This is our first CHO. It is hydrogen and oxygen, and it has a special ratio here, a 1 to 2 to 1 ratio, which means that there's twice as much hydrogen as carbon and oxygen. We have 3 different classes. We have monosaccharides, which means single sugar. We have disaccharides, which means 2 sugars, and polysaccharides, which is many sugars, so like 4 or more typically. Our monosaccharides are going to be the simplest of sugars. Glucose is a great example here. You have different classes, whether it's a pentose sugar, so a 5-carbon sugar, or a hexose sugar, which is a 6-carbon sugar, such as glucose. And you might hear that when we're talking about blood glucose, it's the blood sugar. So here's some examples of our monosaccharides. Notice that they all end in ose: glucose, fructose, galactose, deoxyribose, ribose, right? We have hexoses and pentoses, and they all end in ose, meaning sugar. Okay, our disaccharides here, they are 2 sugars put together. So for instance, sucrose is an example. Our disaccharides here. Polysaccharides, again, are going to be much larger. They're formed from dehydration synthesis, and it's a lot of monomers put together. We have the 2 most important ones are going to be starch and glycogen. So starch is a carb storage mechanism for plants, and then glycogen is what animals use to store their extra polysaccharides. So here's kind of like a little bit about how you make those long chains. Okay, so lipids are going to be our next one. So I said CHO, CHON, and CHON-P. So our lipids here contain C, H, and O, occasionally P, but we're going to just remember this is CHO, CHO. It contains carbon, hydrogen, oxygen. And there's no special ratio here, so that's a huge differentiation between carbohydrates and lipids. Okay, so we have a lot of different types of lipids here. Lipids are extremely important. Phospholipids is going to be like one of the biggest things that we talk about. We have triglycerides or neutral fats. We have phospholipids. We have steroids. And then we're not going to talk about the last one, so don't worry about it. Okay, so triglycerides or neutral fats, these are fats and when they're called fats when solid and oils when they are liquid. Okay, so think about like fats that you might cook with, right? They have different states. We'll talk about whether they are saturated or unsaturated in just a second, but the function of our triglycerides is to help us store long-term energy. I think lipid, long-term, whereas carbs, sugars, that's very short-term energy. It's very quick energy. Lipids store long-term energy. So basically triglycerides, tri means 3, right? So it's 3 fatty acid tails that are bonded to a glycerol backbone. Saturated fats contain as many carbon and hydrogen bonds as possible, meaning that there are no double bonds occurring anywhere. So at room temperature, these are solid, so things like butter. Okay, unsaturated fatty acids are going to be things that have carbon-to-carbon double bonds, which means that it has fewer hydrogens than the molecule is capable of holding on to. So it is unsaturated because it is not saturated in hydrogen, where it has a double bond, carbon-to-carbon bond instead. These are considered more like heart healthy. If we're talking about the omega-3 fatty acids, and that's also really good for your eyes, these are going to be liquid at room temperature. Trans fats are not natural. They are man-made, and they're extremely unhealthy for you. So here's some examples of our lipids. We have a glycerol backbone with 3 fatty acid chains, which is a triglyceride. We have phospholipids, which we have a phospholipid bilayer around all of our cells. So we are made of a lot of phospholipids. These are modified triglycerides because they have a phosphate head on them. So they have a head and a tail. This allows us to create a hydrophilic and hydrophobic region. The head is polar. It's attracted to water, whereas the tail is nonpolar and it's repelled by water. So that allows the orientation of our bilayer to create like a gradient in order to control what comes in and out of our cells. This is what an example of a phospholipid would look like. So it has a polar head on it, and the tails are our hydrocarbon tails. Steroids are going to be really important. We have them in our cell membranes, and they also function to do other things in our body. They consist of 4 interlocking ring structures. The most common are cholesterol, which I said it's on our phospholipid bilayer. It helps to maintain membrane fluidity, vitamin D, steroid hormones, and also bile salts, which are important for digestion. The most important steroid is cholesterol. It's the building block for vitamin D, steroid synthesis, and bile salt synthesis. And like I said, it's really important in the structure of the cell membrane because it helps to maintain membrane fluidity, which means that your cells are not very rigid. They're very fluid. That way you have things passing in and out as they should. So here's an example of a steroid. Okay, like I said, this one we're not going to talk about all that much, but it does play a role in blood clotting and controlled blood pressure. These are two things that have to do with obviously the cardiovascular system, which this class doesn't cover. That's the second half of anatomy and physiology. Then we have proteins next. So I said cho-cho-chon. This is our chon. Protein ends in the letter N. Chon ends in the letter N. So we have carbon, hydrogen, oxygen, and nitrogen here. Occasionally, you'll see sulfur and sometimes phosphorus. It just depends on the different amino acid that we're talking about. So proteins are composed of amino acids. They comprise a lot of our cell, about 20 to 30%, and they have a lot of really important functions, such as enzymatic functions to help us speed up our chemical reactions. They're also involved in muscle contraction, and we have structural proteins, which help to build parts of our body, right? So these are polymers of amino acids, and there are like 20 different amino acids. So there's 20 different side chains, which I'll talk about in just a second. And with proteins, you have several different structural levels, and we're going to talk about those as well. So there's 20 different types of amino acids. They're joined together through a peptide bond. So proteins can also be called polypeptides. Poly means many, peptide is the name of the bond. So that makes sense. If we have like beads on a string, all of the amino acids would be like the beads, and the string in between them would be the peptide bonds. So polypeptide and protein are the same thing. We're going to talk about the different R groups here in just a second. So the basic structure of an amino acid is this like circular part here. It's the amino group. Okay, the R group, the little square, that R is not an element. It is a placeholder because there are, as I said, 20 different types of amino acids. The R group is different in each of those 20 amino acids. But the part that's in the circle, the amine group, that does not change. You have the amine group and you have a carboxylic acid group on the other side there, the left and right sides. The circular part does not change from amino acid to amino acid. The R group will change. Again, R is not an element. It is a placeholder to signify this is an amino acid. It just is an unidentified amino acid. So it's a building block to a protein, but we don't know which of the 20 it actually is because the R group is not given. Okay, amino acids are linked together by peptide bonds, as I said. So you have peptide bonds that form between the two amino acids. It forms between the carboxylic acid group of one amino acid and the amine group of another amino acid. And here's an image of that. We have 4 different levels of structures. So the primary structure is going to be just the amino acid chain. So when proteins are created, you create an amino acid chain. The secondary formation or the structural level here is going to be alpha helices and beta sheets. So based on whichever of the 20 amino acids are present, you're either going to form an alpha helix, which is like a coil, like a spring, or a pleated sheet, a beta sheet. So that's kind of like an accordion, okay? Then we have the tertiary structure, which is how our secondary structures interact. So you'll have multiple alpha helices and beta sheets like forming together. And again, this is all based on whatever amino acids are present on the protein. And then you have your quaternary structure, which is how 2 or more different polypeptides are going to interact with each other. And these form more like globular proteins, which we'll talk about in a minute. Okay, primary structure is just the amino acid chain. There's no folding. It's just a straight, like I said, beads on a string, and it's just a straight chain. Here you have the secondary structure. So you have alpha helices that look like a spiral and beta sheets, which looks like a pleated sheet, like an accordion. Then we have our tertiary structure here, which contains both alpha helices and beta sheets like interacting together, starting to fold up a little bit more. And then our quaternary structure is when we have 2 or more polypeptide chains interacting with each other. We have 2 different types of protein shapes. You have structural, which is also called like fibrous or fibrous, depending on how you want to say it. I like to call them structural proteins. They are strand-like, they are water insoluble, and they are stable. Most of them have tertiary or quaternary structures. Those are your more like complete proteins. Okay, these are things like keratin and elastin and collagen, which is the single most abundant protein in the body. It's really important for us. And we have also contractile fibers, which we're going to talk about actually in our next chapter. Okay, the second type of protein, it says that there's 2 here. Globular, which are functional proteins. These are more things like your enzymes, which are very important, right? So these are more spherical, water soluble, sensitive to environmental changes, which is largely important. Okay, like I talked about, the pH has to be exactly the correct pH for an enzyme to function in. So this is they're very sensitive to environmental changes like temperature and pH changes. These are typically tertiary and quaternary structures again. They have very specific functions, and that's due to their active site. It acts like a lock and a key, where only one lock will work with one key. You can't have like, my car key will not open your house. Your house key opens your house. It's a very specific active site. And some examples of our globular or functional proteins are going to be your antibodies, hormones, molecular chaperones, and enzymes. And enzymes is where we're going to focus a lot. Denaturation is how you unfold or lose functionality in a 3D shaped protein. So these are more of our like tertiary and quaternary folded proteins. So our fibrous or structural proteins are more stable. Active sites become deactivated and denatured very quickly. Those again are our globular, mostly our enzymatic ones we're going to be talking about. Okay, those are our functional proteins. So like I said, temperature and pH are going to be like huge sensitivity factors for our functional proteins here. Okay, if the damage is done, it can typically be reversed, usually reversible if normal conditions are restored. But the longer that you have something that's out of whack, where the pH is incorrect or the temperature is incorrect, the more damage is done. Sometimes you just have to break it down and start over. It's irreversible if the changes are extreme. So for instance, you know that if you boil an egg, cook an egg, hard-boiled eggs, you're cooking an egg on a pan, the white part that's like clear initially turns white, right? That you cannot undo that, right? It's completely a chemical change that has occurred there. You've denatured those proteins. They're no longer going to do the job that they're supposed to. Okay, enzymes. Globular proteins that act as a biological catalyst. A catalyst is something that speeds up chemical reactions. How does it do that? It makes the reaction occur faster because it reduces the input of energy. So it's called the activation energy. So an enzyme functions to speed up a chemical reaction by reducing the activation energy. This means that you don't need to build up a whole lot of energy in order to start a reaction. You only need a little bit to start a reaction because the enzyme is present. Yes, in most cases, you will still have the reactions occurring, but the enzymes make it happen very quickly. So here's some characteristics of our enzymes. Some enzymes come with cofactors, which are metal ions, or coenzymes, which are other organic molecules, typically vitamins. They are very specific because they have a specific substrate. That active site only bonds with a specific substrate. Like I said, lock and key kind of mechanism. And they all typically end in A's. ASE. I like to say that enzymes kick some ACE because they help to speed things up in your body. So like hydrolases, oxidases, amylases, they all end in ACE because they kick some ACE, right? So they lower the activation energy. Like I said, that's essentially how they're able to speed up our chemical reactions. As I said, the chemical reaction is going to occur anyway, but it will occur much faster and much more efficiently if an enzyme is present. So here's some examples of what this would look like in the body. So if you have the graph on the left, which has without enzyme, you have to build up a lot of activation energy before you can actually proceed with the reaction. You see that the reactants and products are there. Then with the enzyme on the right, you can see that the activation energy, that like hill, that mountain to climb up, is a lot less. Like imagine climbing Mount Everest versus walking over a speed bump. It's a lot less. So it's going to happen a lot faster, yet the reactants and the products are identical. So you're not changing the input or the outcome. All you're doing is changing the energy that has to be built up and stored before you can actually start the reaction. And here's an example of how our enzymes function. So we have an enzyme, it's the purple thing, the active site, very specific. It attaches to a substrate. This is an enzyme substrate complex, and this is a temporary binding. Then the reaction will happen within the enzyme. And in this case, we have two monomers that we're putting together as a polymer. So it says peptide bond at the end, which tells you that these are amino acids. So it's showing you here that we have a water molecule being released, which means that this is going to be a dehydration synthesis because we are taking two things, making one larger thing, and we're removing water. So this is the enzymatic reaction happening in this picture. Lastly, we have nucleic acids. So we had CHO, CHON, and CHONP. So CHONP, CHONP, carbon, hydrogen, oxygen, nitrogen, and phosphorus are the largest molecules in the body. This is going to be your DNA. This is going to be your RNA. Okay, nucleic acids are polymers made up of nucleotides. A nucleotide is one piece of a nucleic acid, and a nucleotide contains three parts. It has a nitrogenous base, a phosphate, and a sugar. So there's two different sugars that we're going to be using and a couple different bases that we're going to talk about for DNA, deoxyribonucleic acid. That's what the Na literally stands for. Okay, we're going to be using deoxyribose is the sugar. And then in ribonucleic acid, again, Na, nucleic acid, we're talking about nucleic acids. Ribonucleic acid contains ribose. DNA is your genetic blueprint. It holds all of the directions to make you you. It is a double-stranded helical model. It contains nucleotides that, again, have deoxyribose as the sugar. That's what the D in DNA stands for. You also have a phosphate group and a nitrogenous base. And in DNA, our nitrogenous bases are A, T, G, and C. You might remember apples grow on trees, gas goes in cars. A always pairs with T, and G always pairs with C. We have two different classes of our nitrogenous base, of our nucleotides here. You have purines and pyrimidines. I'm an Aggie. I went to Texas A&M, so I like to think about it like this. Aggie, A-G, right? Our rings are made of pure gold, purine, Ag, purine, ag, pure gold. Maybe that'll help you. The other one, pyrimidines, are cytosine and thymine. Okay, I told you this, A always pairs with T and G always pairs with C. This is called Chargaff's rule. Okay, here's a picture of what your DNA looks like, and you have all of the nitrogenous bases color-coded there. Okay, RNA is going to be a little different here. It is also a nucleic acid. It links DNA to protein synthesis. So essentially, DNA is trapped inside of your nucleus. If you're a eukaryotic cell, the DNA information needs to get to the ribosome in order to create a functional protein. RNA acts as like a messenger and then also helps us to build our proteins. So RNA is a single-stranded linear molecule that is most active outside of the nucleus because DNA is stuck inside the nucleus. RNA is your kind of middleman. It's the go-between. It contains ribose sugar instead of deoxyribose. That's why it is an R instead of a D. RNA ribonucleic acid versus deoxyribonucleic acid. Also with our nitrogenous bases here, thymine is replaced with uracil. So A will pair with U for uracil when we're talking about RNA only. Okay, we have three different varieties of RNA. We have messenger RNA, which is mRNA, transfer RNA, which is tRNA, and ribosomal RNA, which is rRNA. rRNA you won't hear about all that often because it's also called a ribosome. It helps to make up a ribosome. All of these are important for protein synthesis. Okay, lastly, we're going to talk about ATP. So ATP is under the umbrella category of nucleic acids, whereas nucleic acids typically store your genetic information. ATP, adenosine triphosphate, is actually our energy source. So ATP directly powers chemical reactions in our cells. It offers immediate and usable energy that's needed by our body cells, and it functions because you can actually remove the phosphate tail. So it's triphosphate is 3 phosphates. So as you remove each of those phosphates, you're actually releasing a large amount of energy that helps to drive chemical reactions. So the structure of ATP is an adenine-containing RNA nucleotide. That's why it's under the umbrella category of nucleic acids, because it's actually like a derivative of RNA. So here's a picture for you. You have ATP, adenosine triphosphate. It's a ribose sugar that has adenine and 3 phosphate groups on the end. So it says high energy phosphate bonds can be hydrolyzed to release energy. So this means that when you cut those regions, so as you remove the terminal phosphate, you're going to create ADP, which is adenosine diphosphate. You're releasing a lot of energy. You can also remove the middle after you remove the outer one. If you remove the inner phosphate, this creates AMP, adenosine monophosphate, which would only leave 1 phosphate left, and that's how that works. And that's exactly what I just said. So ATP, like I said, can be hydrolyzed to create ADP and an inorganic phosphate, which releases a lot of energy. This is how ATP helps us do cellular work. So ATP drives molecules against their concentration gradient. It also works to do mechanical work such as muscle contraction. So ATP phosphorylates contractile proteins in your muscles so that the cells can actually contract and shorten so you can like flex your muscles. And then to do chemical work, ATP phosphorylates key reactants, providing energy to drive energy absorbing chemical reactions. And that's going to be it. I know that this was fast, but our biomolecules you have been doing since freshman year. Make sure you go back and review basic chemistry if you need to. And if you have any questions, please let me know.